About 2 years ago, we had a conference where one of the speakers was focused on lipid nanoparticles related to the mRNA technology. And she shared some really important information. But as usual, because we only had a very tight time slot, some of those concepts really weren't fully grasped. What she's done is she's spent the time going back over the research, and she now believes that the information and the analysis that she's about to present is going to shape the foundations of the mRNA technology. So this is beyond even COVID vaccines. I don't know what she is going to say, but I am truly fascinated to hear more. So without any further ado, let's just add to the stage Christy. Hi, Christy. Thank you for joining me in this very interesting discussion. So before we start, I want you to introduce yourself to the audience if you can.
Thank you. Thanks for having me, Dr. McMillan. My name is Christie Grace. I'm a former biotech process and project manager. I worked in custom biotech manufacturing, and I worked with teams that custom designed things like recombinant proteins, fusion proteins, and other biologics for large companies that had an idea in their head of something that they wanted made, but they didn't know how to get there. So I worked with teams to design things in research grade and things that would go to clinical trial that are treatments for genetic disease and cancer. So I've got a master's in science. I actually have 5 majors, but you were gracious enough to have me on the last conference, but we only had 15 minutes, and I think I went through 77 slides.
Yeah, so this is now where we're going to take time and we're just going to talk through what it is that you have found. But Christy, you must know that what you may be sharing is huge. Why are you risking talking about it at all?
I need help. Number one, there's some validation that has to occur. Also, the information was kind of buried, and I didn't write a paper on it. And so now I've written a paper and I have submitted it, and it's in preprint form, and I need to submit it for peer review. But regarding risk, I don't see any. I've been frightened before to speak out and was worried, and others have joined in speaking out, including you. So thank you. And the more of us that speak out and present science on the concerns we see that shouldn't be a left or a right issue and shouldn't be religious, and shouldn't be political, is to help people understand things that are going on that aren't being shared with them, and to promote public health and global health. That it shouldn't be dangerous to question things and say there's a problem that we need to address and no one's addressing it. So thank you for having me on. That's what I hope to do today, is to get the attention of people to look at this.
Well, I'm just being clear in terms of my reflection because I still don't know what we're going to talk about. But I know that you know that this is something that's very important. This is a billion-dollar industry. Shareholders, if you are right, and these things are that significant, you could potentially be involved in taking down a whole industry that is now having global reach.
Yes. Yeah.
Wow. Okay. So without further ado, let's get into the science. So you'll start and you just tell me when you want to move along, and I'll move along these images. So here we go.
Thank you. I put together a slide deck, and we're going to go over some basic science. I know it might be difficult for those without a science background to grasp, so I tried to simplify things and start off. So the information that I presented was, again, at the conference, and then on Twitter, I had some posts and threads 2 years ago as well that got I think 150,000 views, but I didn't describe it in the way that I needed to get people's attention, to share what's going on in the modified RNA lipid nanoparticle platform. So what we're going to be looking at are what are called adducts. And I'm going to describe some basic science behind it, what an adduct is, how drugs in the market that caused adducts to form in the human body in the past have been pulled, like Zantac, and what we need to do for a call of action to get others to test this in a lab to confirm, because it's already been tested in the lab with the RNA platform itself, but we need to test humans immediately, starting or with cow blood. So I put this presentation together, hopefully that people can understand a little bit more, so their eyes don't glaze over with science.
All right, let's get through the first thing here.
Okay, so when we talk about adducts and what a genotoxin is, a genotoxin is an impurity that can cause damage to our DNA, and it doesn't have to mutate the DNA to cause the damage. This is a process that can happen over time, but when we talk about pharmaceuticals, there are regulatory guidelines in place called ICH-M7. This is an international guideline that was set by multiple countries, and that the FDA in the United States also recognizes, the EU and Australia, and they are set amounts of chemicals that can be in pharmaceuticals and drugs that we take, and they're not supposed to be exceeded, that are recognized, that are either in the drug itself when it starts off, or when it gets into your body might make it into something else, and then it ends up being harmful to our genes. And that could lead to disease, not just cancer, but you could get diabetes, autoimmune disorder, or other concerns. Next slide, please. So talking about genotoxic impurities, they can lead to mutations. They don't need to insert themselves into the genome, like the DNA plasmid contamination that's been talked about. And it can come from, like I said, a chemical degrading while in the human body, or degrading while the drug's being stored before it's given to the person, or through storage. And these genotoxins can be impurities, and that's what a lot of them are.
Can I just quickly clarify something? Because there will be a lot of news recently. I heard, I think, like Kevin McKernan was talking about plasmids and so on in the COVID vaccines. Is this what you're making reference to, or is this something different?
This is something different.
So this is on top of the fact that they are noticing impurities, say, in the COVID vaccines, for which the regulators have said it's not enough for them to be concerned.
That's a great question, Dr. McMillan. So there's two things happening there. Number one, the FDA and other regulators are stating that the levels of DNA itself does not go over the threshold, or we know that McKernan and Dr. Speaker and Jessica Rose, they just published, I think, in Taylor and Francis for the peer review paper, confirming that it's, I think, over 600 times over the regulatory amount. That's just talking about the DNA. But that biotech plasmid isn't the same as the DNA that was tracked in when we talk about vaccines in the past, and it wasn't in a lipid. So the ICH guideline is something different. And I think that's why I can't state intent. If the regulators would acknowledge that that's a genotoxin, the DNA plasmid itself, then that would also fall under these ICH guidelines, which is different than the amount for the DNA plasmid, if that makes sense. The DNA amount is different than what's called the international guideline for genotoxic itself.
One interesting question that I remember coming up, I'm trying to remember which interview it was that I had, but it was pointed out that part of the problem for regulators was that they didn't have the expertise in this area to be able to understand what was a risk. Is that a fair thing to say in terms of their defense?
That's an interesting question. Vaccines are not the same as biotech, and they were conflated. And we also know there was a video floating around, and I forgot who stated it at a conference, where it was admitted that the modified RNA and lipid nanoparticle platform, which is biotech, was labeled a vaccine because they were concerned about the public not wanting to take it if they said it was gene therapy. Regarding people that are sitting in positions in regulatory agencies, they're very intelligent people. They've studied things for a long time. But vaccines are different than biotech. Legally, I can't speak to intent or to say that they didn't know. I don't have a PhD, and I've been sounding the alarm. Kevin McKernan is a bachelor's degree. He's been sounding the alarm. I mean, you're a doctor, but it's difficult to excuse what's happened here.
Wow. Okay, let's move on. Should I move on to the next slide?
Please. So again, when I speak to ICH M7, it is international regulatory guidelines that are set for the agreement by multiple countries that speak to what are called mutagenic, that can harm our genome, impurities in pharmaceuticals, and the amounts that are set, that are agreed upon, can actually be lowered. It's not, I guess it's not set in stone, but these guidelines are there to protect human health. So I think it's really important with everything we've been talking about and in the news and people presenting to look at these guidelines, because I don't think they were brought up previously. It was just amounts of things where we know, like Wafik El-Diri has talked about the paper that he wrote about where they found the spike protein was interacting with P53. That's part of an oncogenic risk. The DNA plasmid, that's part of a risk. And then what I'm going to present today is really part of a risk, and it's fallen directly under these guidelines in the past. So that's why it's important.
So before you move on, I'm just trying to, because these are such complex topics, and I'm trying to think about how to simplify the concepts.
Okay.
So when you're making, say, the vaccines, and you're using, say, bacteria to help to make some of the proteins or the RNA or whatever, you can have contamination in the product, but as long as it's not above a certain level, it's acceptable. Okay, something like that. What I'm trying to understand is that I get the sense that what you are talking about is not the usual things about DNA plasmids or SV40 or P53, anything like this contamination. This is something that is very specific to lipid nanoparticles. Am I correct? Or is it more to do with the COVID vaccine?
No to all of those. I think there has been the DNA plasmid right now. What's being spoken about is setting precedent and not something that's been really talked about publicly in the past. Same with the spike protein. The ICH M7 regulatory guidelines have been used in the past to assess drugs that were currently on the market after they were already on the market, where it was found that impurities caused genotoxic risk. So what I'm presenting today is going to be speaking to an impurity that's in the modified RNA lipid nanoparticle platform, regardless of spike protein, regardless of DNA plasmid, that has a previous precedent set for these guidelines and these adducts forming that may be a genotoxic risk and a call for alarm to possibly, like Dr. Malhotra and others have called for a moratorium on this whole entire platform. So it's connected to the past, but I don't think anyone's checked this in the human body. They've only checked it in the lab. So I'm going to present the data of what was checked in the lab.
Okay, and so moving on with this here. So this is forces, different types of bonding.
Right. Okay. So I want to give just a little, I want to say science lesson because we're just over video. These are things that are taught in school that we're taught in basic chemistry. Our molecules in our body are moving around in nature, and we have different forces of bonding when it comes to charged particles. For instance, table salt is a combination of sodium, which has a charge of +1 on it, and chloride, which has a charge of -1. And those things bond together. And that's called an electrostatic charge. And this is silly that I have like a butter knife here, but if I would think of this might have one charge, then it's just a butter knife. And if I put a magnet on it, this would be an electrostatic charge. And it's not permanent. I can pull it off and it's not there forever, but this is just kind of simulating what positive and negative charges are attracted to one another. But certain charges are like Velcro, where you can undo them. And in our body, there's things that are moving around in our cells that are moving around, charged particles. I want to go back to the slide, please, because like we said, electrostatic, so kind of on the bottom, if we had two positive things together, those would probably repel one another. Two negative things, they can repel one another, but positive and negative attract. And those aren't always set in stone, right, Dr. McMillan? Like we can have what's called ion bridging and there's shielding, but that's higher level science. But I just wanted to talk about charge because this is important when it comes to the lipid nanoparticle and what's going on inside. Next slide, please. So adducts. Adducts are formed when a covalent bond happens. And a covalent bond is the strongest bond that's possible in nature. And they're happening in our bodies from things like smog, environmental pollutants, smoking cigarettes. An aldehyde can form a covalent bond in the human body, but that's over time. If people that smoke long term, these adducts can form, and like the, like if I show like the butter knife, like the magnet sticking to it, a covalent bond, you're not going to be able to separate that. That's permanent. That's there. That's not going anywhere. Whereas like if it's electrostatic, it can be separated. But a covalent bond is a chemical reaction that is beyond what positive and negative charges do together, that kind of like come like Velcro. A covalent bond is super glue. And it's where chemicals will attach or could attach to our proteins in our body, the RNA in our body, or the DNA in our body. And again, it's like super glue. And we're exposed to them over time, but our body takes time to, sometimes it can usually heal. Next slide, please. So protein adducts can form. Proteins aren't directly involved in cancer, but there can be cancer risk. Another thing that can happen is adducts can form with chemicals, environment, pharmaceuticals, where they're permanently binding together, and then it can change the way that a protein folds, because we just have, Dr. McMillan, how many proteins do you know how many proteins we have in our human body?
I have no idea.
I mean, just insulin is a protein.
Yeah. You mean how many types or how many proteins?
Yeah. I think the liver makes like 1 million a day.
Wow. Wow.
Motor proteins. Motor proteins are if anyone's watched a video, there's a little ball with legs like walking on like a tightrope in the human body. If someone takes levothyroxine for their thyroid, part of that drug is cleaved, and then the motor protein carries it, kind of like, it doesn't have a back, but on its back, and it takes like 6 days to get to the cell. But those are proteins. Our proteins in our body have functions to keep us alive.
Mm-hmm.
So proteins can be adducted, can be formed adduct, and what can happen is, our immune system might recognize, depending on how many proteins have something super glued to it, that say like a chemical, can cause an autoimmune reaction, because the body will say, what is that thing? That's not my normal protein. Why is that there? Depending on the amount or what's going on. And that can have like a cytokine response.
Yeah, so that once you've mentioned that, that makes me think about the work that I was doing in autoimmunity, and the spike protein being like Velcro, sticking to normal proteins, and then the body recognizing that as an abnormal protein and therefore causing autoimmunity kind of mechanism when that kind of bonding can occur.
Yeah. Yep.
Okay.
Next slide, please. So RNA, I think modified RNA, when we think of the modified RNA lipid nanoparticle platform, RNA in our body makes proteins. We start off with DNA. DNA makes our RNA, and then RNA is the blueprint for proteins. So these can also be, RNA can be modified too. And again, it would be something sticking to it like super glue. If a chemical sticks to the RNA, which has 4 letters, and if we think of RNA like letters in a book, there's letters in RNA. DNA has letters in it too. But it would alter the property. It can alter the property of the protein it's going to make. It might make a useless protein, or it might make one that's a junk protein. We'll talk, I guess we could talk about that in a second when we talk about the N1-methyluridine. But similar to that, where something's happening and getting stuck to the RNA. So then when it goes into this thing called the ribosome, it's kind of like it's like a thing in the cell that reads it and kicks out the protein that it makes from that, the blueprint. If the RNA enters the area that the cell is supposed to read it and translate it, and it has something stuck to it, that can cause the same kind of frame slipping.
And so let me just make sure. So the adduct, as you're describing, is any chemical that superglues to either a protein, RNA, or DNA.
Yes.
And whenever it does this, it can change the function and the way how that either protein works or the RNA is translated. So in effect, an adduct stuck onto RNA can make an abnormal protein.
Yes.
And
depending on where it is. Yeah.
Yeah, an adduct stuck onto a DNA can make an abnormal RNA, which would then make an abnormal protein.
Or worse.
That's where the cancer. Okay, good. Making sure we're getting it.
Yeah. Next slide, please. All right. So in the past, there have been drugs that have been pulled off the market because they bound to DNA like super glue. And is it carboplatin? I pronounced that wrong.
Yeah.
That was a drug for cancer, right? To help fight cancer in people. But what was found after the drug went on the market is that this drug meant to fight cancer got stuck to a piece of part of the DNA in our body. So super glued to part of the DNA. And so when the body is reading the DNA, it could break it. It could break the DNA. It might not just be stuck to it. Same with the RNA. It could break it. So it's now snapped in half. Or depending on what gene in the DNA it's super glued to, that could be P53. That could be a tumor suppressor gene. That could be an oncogene, and it could interrupt the function of that gene. Not necessarily making the RNA, but it could interrupt the function of that gene in the human body. And so the same thing, if anyone's heard of Zantac, that's why Zantac was pulled off the market, because they found that Zantac, the it's not a proton pump inhibitor, but it's like, isn't it H2 blocker?
H1.
H1 blocker.
Yeah.
It was people took it because they had nausea or stomach pain, and they were taking Zantac. Was it ranitidine?
Ranitidine, yeah.
And it was after the fact that people started doing studies and found that as the body's breaking it down, the drug formed a secondary thing, what's called like an intermediate, where it formed another substance that is gluing itself sometimes to the DNA in our body, and they found it to be carcinogenic, genotoxic, meaning harmful to our genes, and that's why it was pulled off the market.
Right. Wow. Yes, that's good to remember. All right. Moving on.
Yes, please. Okay. So tobacco smoke can do the same thing. I think aldehyde, I'm trying to think of the chemicals that are present in tobacco smoke, and over time that can cause DNA damage. And not everyone gets cancer from smoking, but it can cause mutations, and repeated mutations can lead to cancer. There's the multi-hit theory of cancer, where if you keep getting mutation in the same spot, or people that are pregenetically disposed, is it Lynch? I'm trying to think of the different predispositions where genetics of your parents can have alterations in your genes. You might have a mutation that, like our body's getting lesions all the time, whether we smoke or not, and our body's repairing them over time, over years. And, I have friends that are Irish Catholic, Marianne, I just said one of their names. They're in their 80s. They drink 2 or 3 Mickeys a day. They smoke cigarettes. They don't have cancer. They're in their 80s. That's just their body's healing it, where other people could smoke and then in their 40s or 50s get lung cancer.
Yes, it's a fascinating thing that, as you said, there is a predisposition. It's about 20%, we would say, who smoke. It's almost as if they accumulate all of the complications. They get heart disease, lung cancer. The problem is you don't know which 20% it is. And so they start to believe that they are fine because they know an 80-year-old who is smoking who's been fine as well. And it's that kind of situation that causes the problem. But essentially, again, part of some of the chemicals in tobacco smoke can bind to particular parts of the DNA. And for people who are vulnerable for cancer, it can then cause cancer to form.
Right. Or they might not be vulnerable, right? Like they might have confounding factors. Other things they're exposed to different times in life, right? Males and females, different parts of the genome. Like we have 2 X chromosomes, where males have XY, and there's different genes on the X chromosome that can have increased autoimmune conditions. And I know there's links, correct, to autoimmune disorder and cancer. So I think there's probably a host of things. But again, so let's go to modified RNA and LNP. All right. So we're going to look at a study that Moderna did by Moderna scientists, paid for by Moderna. Their names are Packer was one of the scientists, Packer et al. So again, Moderna scientist, Moderna lab, paid for by Moderna, and it was published in 2021. So we're going to look at the RNA. Like we know the modified RNA. Dr. Tay and Dr. Ko out of Korea, I think their paper came out in 2020 or 2021, where they looked at the storage conditions of the RNA and LNP, and that it was degrading during storage, and it was breaking down. And they were finding that the RNA was fragmenting, so pieces of it. But they also found that the lipids were sticking to the RNA sometimes, and they weren't just electrostatically binding, like where I showed, like you can pull the thing off of the butter knife. They were like super glue binding onto the RNA.
And can I just clarify here, just so that people get this? So on your left here is one lipid nanoparticle, for which it will have these I guess, phospholipid-like materials on the outside, making it into this ball. And then on the inside, it will be protecting the mRNA, the modified RNA, which is then packaged and taken into the cell. You're saying that even within this lipid nanoparticle, some of the RNA binds to bits of the lipid nanoparticle as adducts.
Yes.
And
Yep.
it will then cause sometimes the RNA to fragment so that it's no longer a full segment of RNA, but pieces of it, which would then make abnormal proteins.
Or not at all. There's been studies that showed that non-coding, so the body can't do anything with it, the body can't read it, can either turn off or turn on cancer. There was a paper that said it can, non-coding, meaning not readable. The body doesn't know what to do with it, and it's just junk RNA, so not making a junk protein.
And so it can't degrade it. Why wouldn't it degrade the RNA that's junk?
Wow. Or it could take time. So we can talk about that in a little bit, because I think that I probably have like 15 more slides.
Oh, all right. Let's move through quickly then.
Okay. All right. Okay. So here's a study. Moderna scientists wanted to know, they did their own study that was different than the N1 methyl pseudouridine study, where they were finding that some of the proteins in the modified RNA for the COVID RNA LNPs were making junk proteins, and they wanted to know why. And next slide, please. So I guess we can go a little bit faster. So there's the abstract. And what they found is they used a method to look at the lipids in the RNA that had never been used before by scientists or published ever. So this was the first time any scientist did this study, and they used what was called reversed ion pair high performance liquid chromatography. And what that is, where they put the modified RNA and the lipid particles inside, all the lipids that were inside, whether it be the positively charged lipids, the PEG, the cholesterol, the DSPC, they put them through columns and they separated it, and then they looked at what was happening with all the lipids that were surrounding the RNA in the lipid nanoparticle, and they found that some were binding permanently, superglued to the RNA. So here's a graph, and if you can go out to the study and see, it's open access, so you don't have to pay for it. So thank goodness for that. Anybody can go find the study. So here's where they looked at time, and they looked at temperature, and they looked at the different lipids. I just want can you just go to the next slide, because I'm going to show you temperature here. So what they did was they took all the lipids and they took the modified RNA and they put them at different storage temperatures, and then they looked at the temperature versus the number of superglue happenings. So there's the adduct. -20 had that's usually what it's stored in, had the lowest number of the adducts forming, where the lipids were binding to the RNA permanently. 40 degrees Celsius had the highest number. And if we think about body temperature, human body temperature is about, is it 37 degrees?
37 degrees
Celsius. And that's quite a jump from the 25 to the 40 degrees Celsius.
And just to clarify, because initially the vaccines were supposed to be stored at -20.
Correct.
But then that changed, didn't it?
Right. They're supposed to be stored in their refrigerators. I think shipped at -80, because as it's shipped, it's going to get cooled down, or warm up rather, and then -20 when it gets there. And then they changed it. And like we know through different reportings out of Australia and other places, that they're storing it for months before they ship and then after they ship.
All right. Okay. All right. So that's important. So what does this mean?
So this is, they used what's called mass spec, and this was just, they were looking at the N1-ME for N1 methyluridine, and they were looking at the lipid molecules, and they were looking at which of the letters on the RNA, there's 4 letters, were getting modified. So this is over time when they were coming in contact with the modified RNA. And this is a whole bunch of, I guess, science, but they were looking at time over minutes. And it's, yeah, A was the highest peak, but they were.
And what does A mean?
So that was the highest number, when they say signal counts on the left, the highest number of adducts that were forming.
And this is the N1 methyl pseudouridine.
Yes.
So this is number 2
Yeah.
in terms of adducts.
Yeah.
So that's important.
So this is the
A,
letters that are A. A.
yes,
Sorry, go ahead.
AC. Okay, yes. So this is important here. So in effect, then, let me try and remember. So A, C, G, and what's the fourth one? U, uridine.
Correct. And
Yes.
in DNA, that's swapped out for the T. Yeah.
Yes. So
Yep.
the uridine in the ACGU, the uridine in the mRNA has been switched to methyl pseudouridine. And so what that is saying is that the methyl pseudouridine, when we look at this, is the one that has the second highest adduct formation behind the A, and A is the adenine.
Mm-hmm.
So adenine is highest,
Mm-hmm.
and then the methyl pseudouridine. G, which is guanine, is third, and then C, cytosine, is the lowest.
It's being modified, meaning it's being altered.
Mm-hmm. Okay. Right. Okay. All right. Let's move on. So yes. So
This is
what
where it's
does
going to
this?
get interesting.
Yeah.
So the researchers found that the adducts are forming. So the lipid particles, which I'll show in a slide in a little bit, are getting superglued in some places because there are impurities in the lipids themselves. So there's 4 parts to the lipid nanoparticle, which is on the next slide, but they found that one of the things that is being formed through storage or just through the manufacturing process as well is N oxide, and aldehydes are another. They found more than one, but that's just the chemical chain and what's going on there. So when we talk about the lipids, they were on the first screen too when we first started. When the scientists saw where these are forming, and they found that the ionizable lipids, there's some that have a positive charge, and these are attracted to things with a negative charge. The RNA has a negative backbone. So inside the LNP, they're kind of around it, kind of like in a layer. And then there's ones that are ionizable, meaning they don't have a positive charge yet, but when they enter the cell, they will. So they haven't really like stuck up against anything yet. And the cholesterol and the PEG, they weren't really finding it was doing much. It was mostly in those ionizable lipids, and those make up percentage-wise half of the LNP. Half of the lipids in the LNP are those. So there's its little adduct, and what's going to get kind of scary is what's probably happening in the human body. But those are just pictures, and they were showing that the lipids weren't interacting with the sugar part of the backbone of the RNA, just the bases, like you said, the A, T, or A, U, C, and G.
Yes.
Next slide, please. So then they also found that these adducts, so the lipids are basically like, this is terrible, there's a little thing hanging out. Like if you imagine like this was the RNA and the lipid is electrostatically bound, kind of like the example I used with the magnet and the knife before. They're bound by charge. Like if you imagine the RNA, DNA, and proteins have negative charges. And if the positively charged lipids come in contact with this, some of them in some spots are getting superglued to the RNA in certain spots, and it's not reversible. So when we talk about the frame shifting and the junk proteins that were found in that study, and I apologize, I forget the authors of the N1-methyluridine, where they said that the N1-methyluridine was cause for frame shifting and making junk proteins, as we just saw on this chart by Moderna. And that was actually done, this study was done before their study, so I'm unsure if those researchers saw this. But because the lipids are getting superglued in certain spots of the RNA, when it goes into the part of the cell that has to read it like a blueprint to make a protein, it kind of skips, and then it makes a junk protein.
Yeah, okay. And what does this show here? So this is the link between the normal protein expression and the adduct or adduction occurring with the mRNA.
Right.
So the protein expression is highest or lowest in the LP, which is the lipid nanoparticle?
So I think that's like there's like a late peak. So there's like a medium peak in control. So this would probably take more to explain than I think we should on the show here.
Let's move on. Yeah.
Thanks. All right. So this is where this gets important. So the lipid nanoparticle doesn't really look like this. They're like little blobs. They're not spherical, but this is important. So Dr. McMillan, we were talking about what's in the lipid nanoparticle in the COVID RNA LNP platform. We know so at the center there, that's the RNA. And we know that there are probably 2.5, 3 per bubble like this. But sometimes there's more or less depending on how it was made. And in one dose, and this is where the math and adducts is going to come into play, in one dose, 43 trillion RNA per dose per person, approximately. On the outside, we've got the peg, that little squiggly thing that they placed there so the immune system wouldn't see it in your body and attack it, so it could go to head to toe, even though they told us initially it was just in the arm. But that kind of disguises it from the immune system. And then the DSPC kind of gives its kind of like skeleton shell, I guess. Right? So it's kind of like interwoven on the outside. And then cholesterol is kind of the bulky stuff, mostly on the inside. But then again, half of this thing is what's called an ionizable lipid. And it's either no charge right now, and when it enters the cell, it's going to get a positive charge, or it has a positive charge right now. And if you look at what's around the RNA, I mean, this isn't an exact number, but because they're positively charged, they're just surrounding the RNA that has a negative charge. And again, that's causing some of them to break because it's effectively mutating or gluing to the RNA.
So a question here. So why is it that, what's the purpose of the ionizable lipids? Because you already have the ball, so to speak, which is being held together by the PEG lipids and I guess some of the chargeable ionizable lipids. Do they serve a purpose on the inside of the bubble?
I think it protects the RNA, and I think it helps with the overall charge too, when it's the lipid nanoparticles getting into the cell, if that makes sense. It makes it get into the, oh my gosh, there's like a few reasons. There's the RNA. It helps with the transfection a bit better, which sounds terrible.
Well, what I mean is that when we look at it here,
yeah,
and we see, so I can imagine, so the PEG on the outside hides it, and I guess to some extent helps it to get inside the cell. What I'm wondering about is, what is the purpose of all of these ionizable lipids around the mRNA inside?
Okay, so that we would have to get to charges and net charge of the LNP. So it helps bring in part the net charge of the LNP to closer to neutral. That's one thing. Okay, so sodium, this is going to get kind of graphic. When I talked about table salt, sodium has the letters Na that has a positive charge of 1. Chloride has a negative charge of 1. And when you put these together, that's a charge, that's no charge. It adds to 0, right? But if you just ate sodium by itself, you wouldn't be alive because it's very reactive. And not that would happen with these things, but It's helping bring the total, part of it is helping bring the total net charge when you add them up close. It's supposed to bring it closer to neutral. That's one of the things it's doing, because Pfizer said in its own data sheets that the total charge was 0, closer to -3 on the LNP, to avoid concerns in the blood compartment, meaning clots. Like, so it's supposed to have reduced toxicity in the bloodstream, basically, because studies have shown if you put something really positively charged into the body, that would be really high positive charge, that could cause clots. Things that could be super negative charge on it could cause clots. So it was supposed to reduce toxicity, was their thinking in the bloodstream, was part of it.
I did a presentation recently thinking about, when I was talking about clots, embalmer's clots. I've been following that for a while. And one of the questions I was asking was, what happens to the remnants of the lipid nanoparticles? How does the body get rid of it? What does it do with them? It's release the mRNA, the cell, it is inside a cell. And what I was saying is that this now becomes like plastic in terms of what happens in the environment, where we have lots of plastic. It's not biodegradable. We have nowhere to put it. We have no landfills for it. Is it a similar thing with regards to these ionizable lipids and PEG lipids inside the cell? Do they just get degraded, or do they have to then be packaged like plastic?
Wow. So, okay, so going back to the positively charged lipids, there's different types of lipids. Part of that is so the RNA can release in the cell, because some of the things in our cells will do things based on charge, if that makes sense. So we have what are called macrophages. If anyone has seen a video of, I don't have another word besides blob, where it's this thing kind of chasing something across the screen, and it's kind of like eating it and then digesting it. Our macrophages can detect the lipids, and part of the PEG on the outside is for them to digest them. And charge, that goes into another topic. If they're eating a lot of charged things, that can lead to clotting in some people, like what happened during Delta wave. There's been papers on that for the first wave, because the spike protein had a really different charge to it. Some of these lipids will become part of the cells. I don't know that that's been placed in literature fully yet, because of this Moderna study, where it's going to permanently bond in some cases. Some of it's supposed to be excreted out through the kidneys and processed as it exits the liver.
Wow.
So there's supposed to be exit pathways, but then when we look at like the pores in the liver, like I'm going to misspeak, the glomerular. There's pores in the kidneys and livers that can only handle certain size things, and some of these lipids are beyond the size they're supposed to be. So all of this, when I was presenting on your COVID conference 2 years ago, I presented this in a different way, where I was saying parts of the lipid nanoparticle were going to cause frame shifting. Parts of the lipid nanoparticle were going to cause junk proteins, misfolding of proteins, could lead to autoimmune, could lead to cancer. And I didn't present it in the way that I should have. So recently, this paper is on the preprint.org, and I explained using Moderna's own study how there's no reason, Dr. McMillan, there's no reason why this isn't happening in the human body, that those lipids, when entering the cells, can create adducts with our DNA and our RNA and our protein. The thing is, when we talk about DNA plasmid and that contamination, this is going to be higher amounts by quite a bit.
Excellent. Right. So let's move on. So this is the paper here.
Yep.
And this is talking about this adduction process with lipid nanoparticles.
Right.
Mm-hmm.
So if we go to the next slide, So it's already known in science that if you get an adduct on your DNA, RNA, your protein, it may or may not do this. It depends on the amount. But through smoking, as an example, we can have things stuck to the DNA in our body that might cause nothing might not. Something might happen, maybe nothing, depending on where on the DNA is and how many are impacted in our body. But we know that adduct formation is responsible for birth defects, diabetes, cardiovascular disease, autoimmune, and cancer. And there are studies proving this.
Okay.
If we go to the next slide, please. This is what some researchers looked at when they found where in the body the lipid nanoparticles go. So this is just a reminder that it's going everywhere. It's going to lymph nodes, it's going to the heart, lung, liver, spleen, bones, brain.
Just in different amounts. And what is SP?
That's just a different spleen. Spleen. LV is liver. Yep, LG is lung. But we know that if there's different charges on the particles, they can go different areas, ovaries, testes. So that's a concern. Next slide, please. All right, so here's why this matters. So we know that Dr. Rose and Kevin McKernan had their findings published in Taylor Francis. So we know that inside the lipid nanoparticle, there is DNA plasmid. And I didn't really bring up their data because we would be just going through so many slides. But they found, and so did Raoult, so did Dr. Buchholz, so did Bridget Koenig. Lots of scientists around the world, I don't want to say lots, but under a dozen, that's a good number, found DNA plasmid contamination that exists because the DNA plasmid is what was used to make the RNA in the E. coli that you talked about earlier. And I think the average size was 100 to 200 base pair, but it was found to be to maybe 200 billion pieces per dose. I think that's something around there. So these DNA pieces are the same as the RNA. Chemistry is chemistry. The same thing that the people at Moderna did not check, but it absolutely, I would stake everything this is happening inside the lipid nanoparticle is that the lipids are also binding. These lipids are also binding to the DNA plasmid in the lipid nanoparticle in the modified RNA platform. So if the DNA plasmid enters a cell, wherever the DNA plasmid goes, these things are going to go. If the DNA plasmid enters the nucleus while the cell's dividing, if the DNA plasmid doesn't integrate, it doesn't mean that one of these positively charged lipids that can bind to our DNA, like we don't know that that's going to do that or not, but it has the capability to, because that is basic science.
One of the things that I keep on hearing from people who will go against what you're saying is that they will say, show us the evidence that this is a problem. We've given billions of doses. If this was a problem, we would see it by now. What would you say to them?
We know the studies that have shown that colon cancer is on the rise, especially the last few years. And colon cancer, I think half of the human population already has a polyp in their colon. So it's a growth in the colon, and the growth is from mutation. No one has shown what I'm talking about in the human body, and Moderna did show it in the lab. And we get mutations all the time, and no one's thought to check. So what I'm saying right now is no one has checked it in the body and needs to.
Yeah, I agree with you. I'm just saying that from what I have seen with many of the defenders,
Okay,
they will say to you, we haven't seen any problems, and it is quite likely that just because it is present doesn't mean necessarily it is going to be a problem. And they almost push the responsibility for you to prove harm rather than for them to prove safety.
they are pushing back.
Yes, I'm
That's
just
true.
telling you, I get it all the time. Is that the
Um,
pushback is prove to us that this is causing harm. So I argue it's a similar thing that happened with the smoking lobby. When there was initially an association between smoking and cancer, their pushback to the scientific community is show us how. And
okay.
they couldn't. And so they managed to deflect that question for almost 30, 40 years just by saying, prove to us that this is the problem.
Money.
I'm
Like
just,
you
yeah,
said in the beginning,
yeah, yeah.
pharmaceutical companies have a big stake in this. Some of it is ignorance, blind ignorance. We know people who lost family members an hour, days after getting one of the doses of this stuff and still denying that it was an issue. Um, I think that when Dr. Macri went on TV or on video, it didn't take him doing that for many of us to know that we all know someone who has had some type of injury from this stuff. But so when you say that, yes, quantifying it, yes. So this, what I'm presenting here, is something that you can quantify in the same way that researchers were able to do with smoking. It wasn't until the 70s, was it the 70s or 80s, when researchers found through smoking an aldehyde formation and using what's called mass spec and other imaging that they found that these adducts were forming, and that is how they quantified it. And the presentation I'm giving here is showing how you can quantify if there's cancer risk, and I believe there absolutely is.
Wow. So when we look at this slide here, what is this?
So when we talk about the DNA plasmid that is in the modified RNA and lipid nanoparticle platform that's left over. A lot of people have said that it can't go into the genome. It absolutely can. So all the slides here came from studies I didn't make, or I didn't make these pictures rather. None of these pictures are something I made. I've got a reference deck for these. So that would look like a piece of plasmid entering the cell, and it can get in through the microtubule and through these pore complexes. One of the things that the positively charged lipids also does, which is just a nightmare thing, is a few fold. Number one, charge can be a nuclear localization signal. It's not, it might be part of the SV40 promoter that's found, but studies have shown that positive charges help and can assist things entering the nucleus, because the outside of the nucleus, even though it's not shown here, is can interact with charges. When we talked about things breaking down, like you've said, people push back, and there's been scientists who have said, "Oh, the enzymes inside of the cell, kind of like our enzymes in our mouth, break down sugar. The cells have enzymes to break down DNA that shouldn't belong there." Well, when you've got a positively charged lipid stuck to it, that's another thing that would slow that down. Beyond the issue of the plasmid DNA being resistant to being broken down anyway, same with the N1-methyluridine. Now this is an addition to that it can help it get into the nucleus easier. And the modified RNA was found in plasmid for up to 28 days in people circulating. Besides the N1-methyluridine, this is a fuzzy green thing, but if you think of it like a lipid, if these are stuck to that modified RNA, it will shield the modified RNA and or the DNA plasmid additionally from being broken down, because the enzymes are actually kind of charged. They're reacted to charge. Otherwise, they'd start eating everything in the cell.
And I think this fits in with the issue with regards to exosomal problems, where these cells now become overloaded with some of these abnormal products, can't manage them, and therefore they package them up into their own little nanoparticles and share them around.
Or go dormant in the cell. And that is a document, if you need, that I don't have included here, that was put up by the FDA and CBER in 2020, that plasmid DNA can go dormant in exosomes and hang out in the nucleus for up to 20 years. and then kind of
Wow.
be reactivated like a virus
Wow.
or do
So what
nothing.
does this slide here mean?
So this is just another one looking at the complex way of, like on the right-hand side, there's a lipoplex, polyplex, plasmid, liposomes. Those are just kind of ways lipids can form with DNA and how that can actually get to the nucleus. If that's a lot of science for someone says no way. So this is just an extra slide showing this.
And it's important to note that once it gets into the nucleus is a protected region, like the cell is a protected region.
Right.
The nucleus is like the vault inside the vault.
Right.
Yes, yeah. So these are pretty serious, very, very serious things. What's this about histones that you've got here?
Oh boy! So I actually showed this in your presentation 2 years ago, and I thought about using the deck. So histones surround our DNA, and if they get damaged, you can have chromosome alterations, aging that could lead to cancer. I'm not saying it isn't everybody, but the histones are proteins that surround our DNA that have a job to do. And again, these positively charged lipids, now these are in each dose. If 43 trillion RNA are in each dose, and when we talk about half of the dose is these positively charged lipids, we're in the quadrillions. And when Moderna looked at a really small percentage of them are making these adducts, we're still into the trillions if hundreds of trillions should react. And also they're going to react more in the human body, but as far as the histones, they can, so once those positively charged lipids enter the cell, through electrostatic reactions, they can covalently bond to things, and if they get in the nucleus, they don't have to integrate. And I know that, and I've heard leaked information that we know McKernan said that he has the tumor that had that plasmid in it, that someone I think died, unfortunately, of colon cancer, but then others didn't find any DNA plasmid. So this is an additional thing that can be happening. And those plasmid pieces, when they enter the nucleus, if they are, and some of these lipids might be entering on their own just through the pore, through charge, if they damage those histones, that can alter the way that our DNA works within our chromosome and how it can create proteins. And again, it can lead to faster aging.
Well, one of the most
Cancer.
Yeah, one of the most important things that I'd found recently was a paper looking at acetylation of histones related to mRNA vaccines in macrophages and locking them into an inflammatory position. Now, that perfectly aligns with what you said in that we don't know the exact mechanism. But I remember when that piece of work came out, it explained one of the patterns, which is that some people can get persistent inflammatory processes like long COVID just from being vaccinated.
Right.
You know, and it seems as though it impacts on those histones. And so if an inflammatory part of the gene is already unwound, it locks it so that it can't get back into position.
I didn't know that. And this might be a driver of that then.
Yeah. So this is the point. As soon as you saw the histones there, I remembered that paper that came out this year specifically talking about they know this happens, they don't know the mechanism, but they can see that pattern. And for me, that then explained the macrophage hyperactivity that we get largely around the pandemic, which seems it can occur from infection. But if you then have infection plus mRNA locking it into position, you can then get persistent inflammatory processes.
I know there's long COVID can do it too. I think there were people that had that, and that was charge based as well. I know this was a lot, and that was the last slide, but I think this is in the top 3 of spike protein, DNA plasmid, and these adducts forming, because we know in the past from drugs that formed adducts in the human body, if it was found, and usually it was after the drug was on the market, and someone went and looked and found they were forming adducts, and it was doing it with our genes, then it was off the market forever time. And the ICH says that it has to be under, I think, 1.5 milligram. And if we calculate the molar mass of this stuff by, like, was it Avogadro's? I said that wrong. Number. We're over that. And so this should be like top of the news. I don't have money to do it. If you know someone who has money to do it, you don't even need it. Sorry, go ahead. You want to say sorry?
So this is the issue now. When you think about it from a scientific point of view, say, for instance, I just did a presentation today which showed that in 2023, a Ramsden poll, I can't remember if I pronounced it properly, showed only 10% of the population thought that they knew someone who was harmed by the vaccine. It's 56% now. And so there is a growing recognition that there are problems, but it comes with a huge cost. Nobody wants to find this out, not even people who may be affected. It's the most uncanny thing, where the biggest push to find answers are not coming from the part of the population who is at greatest risk, but it's from the population who is at least risk, in the sense that it seems to be the unvaccinated people who are pushing for this kind of research. Now, it may just be that they want to be right, in the sense that, you know, they went through a lot and they want to prove a point that they were right. But for the vaccinated cohort, they just don't want to know. How do you get around that?
That's difficult. Dr. McMillan, I'm interested in your thoughts on that. All right, so I remember my ethics classes, if someone positions themselves on the side of an argument that research has found or studies have shown that if you're given more data, people will more often than not try to use that data to strengthen their argument and dig their heels in, and they'll find a way to do it. It's just sometimes human nature. To still not take it in, that you could tell them and show them all the data and they'll still just deny it. And I think part of that is identity, and that is it. Would you say ego, a bruised ego, or fear too? Fear, I mean, of knowing.
Fear, yeah. So I think there are 2 points to it. It's kind of like what happens with a diagnosis of dementia, or in the past with a diagnosis of cancer, or even before they had the therapy for HIV, the diagnosis of HIV. There's nothing I can do about it. What is the point of telling me if there is? So if you have something that I can do that can protect me, that's different. But if you are just telling me for the sake of me knowing that now I'm at higher risk of something I can't do anything about because of something I was told, they don't want to know. Or let me phrase it another way. Let me ask you a question here, Christy. Have you ever been scammed?
Yeah.
Do you talk about it?
No. Embarrassment.
Isn't it important?
Low to the ego. Embarrassment.
This is what I'm saying. I too.
Shame.
And so people don't like to acknowledge those issues. And even though there may be harm, they would rather to protect those other things. So you're not just up against pharma industry. You're up against the fact that the vast majority of people don't want to know.
I agree with you. I also think, and what I just presented here today, which is the first time I showed this like slide deck, needs immediate attention. You can check the numbers. You don't need to get vials of the specific product. You can order these lipids and pay for them. Even if you were using blood, I don't think you don't necessarily need human blood either. Use bovine, even if you want to start there. There's a lab nearby, but I think this is extraordinarily significant, and it needs immediate attention because it would align with ICH M7 guideline, and it's not part of the DNA levels that we're getting pushback on. This is an international guideline. Moderna showed adducts are forming. It's Moderna paid for, not anybody else. It was Moderna that showed this data. And there's no reason, I'm unsure anyone else or your opinion on this, there's no reason through science and molecular biology that we know it, that this isn't happening in the human body at a very large amount.
So
and you're getting it in one shot.
Yeah. So here is a practical final question as we close out, Christy. How is there any way easily? Because don't expect learn from the tobacco industry. They will fight to the very end to protect their interests. So you can't assume that industry will do the right thing. But for the individual who may be listening, what can they do that would be able to potentially check their own personal risk?
I'm not a medical doctor, so I can't diagnose or treat disease.
But is there a blood test? Is there?
For this, if people had DNA tested before, some of these modifications that could have been made, when we talk about the reactive oxygen species, there's a gene, oh gosh, it's like a OX8 gene that can be mutated just through the reactive oxygen species. I'm not saying that anyone should get their tissue tested or blood tested, and I've heard this before. This is a plea to scientists to say, I think for me it would take about $5,000 to $10,000 for me to check this at the lab here that I could rent out. As far as a person goes, we've already had other people raising the alarm, like you said, and there's not a lot of tests that can be done. So I think a group has to come together, a larger group of people.
So let me just get this. So I want you to, as a closing, Christy, I want you to make the call. Okay? I want you to pretend that you just say what it is you need, everything you need, the amount, the resources. Just pretend for a moment that anything you wanted, you could get. Your last call to the world. Tell us what you need.
To myself or scientists, for me, the lab here, I have some strapped finances right now. I wouldn't even put it in my bank. It would go to the lab. There's an extension of the university lab, full biotech lab. It's,
don't go to the
3,000.
ocean with a spoon here.
Okay.
No ocean with a spoon. I want you to go with a bucket.
One person, one scientist. Take the lipids, mix it with blood.
We want more than that, Christy. We want, suppose you wanted to do it on 10,000 people. This is what I'm saying. You have no limitations. Make the call.
Get Jay Bhattacharya and RFK in the room right now with a group of people.
You want them. You want to be part of NIH? You want to do this research through NIH funding?
Yes. Yeah.
How much would it cost?
Under 10,000 if we just do the blood.
No, but that's just for
Phenomenal.
one. Let's just talk about if you're doing it 4,000 or 10,000.
Blood. We're just talking about blood samples and the equipment. We're not talking about injecting into tissues or animals or anything. We're just looking for the adduct formation in cells.
And this is just blood samples and lab.
Yeah.
Wow.
Yep. Just put it under the HPLC. Just put it right under there.
So anyone who is listening to this, who has access to RFK Ray and Jay Bhattacharya or anyone else in these organizations, somebody with the expertise to know how to prove this, says it costs a fraction in the context of the benefit and the knowledge that will come, not just for the USA, but for the whole world.
Yeah.
Thank you very much, Christy. And I wanted you to make that call to the world because you never know who is listening.
Okay. Thank you.
You never know who is listening. So
Okay.
yes, this is what we are after. And we hope we give you the license to find the answers for us. Okay. So thank you very much, Christy. We're going to close now, and I want you to wait for me until after the outro. But thank you, everyone, for watching. This was complex, heavyweight stuff, but we really appreciate Christy coming to try and dissect the science for us. Have a great evening.
Thank you.